So in part 1, we saw a brief introduction to glycolysis, why it is happening, what is happening. We saw that it is a part of cellular respiration, and we also saw some of its salient features. We saw that one molecule of glucose undergoes glycolysis to form two molecules of pyruvate. What is basically happening is that glucose, a 6-carbon molecule, is getting broken down into two 3-carbon units of pyruvate. And this reaction of a 6-carbon molecule getting broken down into two 3-carbon molecules is taking place in 10 steps. In these 10 steps, there forms several intermediates whose knowledge is essential for learning and understanding glycolysis. So let's have a look on the general idea of their structures and nomenclature. We have first the 6-carbon intermediates. Very simple. We will take one C double bond O unit and 5 COH units. So we have 6 carbons in total. We will arrange these 6 carbons in a straight chain 1, 2, 3, 4, 5, 6. And now, based on the position of C double bond O, we have two isomers as our two intermediates. In first, we will place C double bond O on position 1, and in second, we will place C double bond O on position 2. All the 5 carbons left will be COH units. We will complete the valencies with hydrogen atoms as required. And there you go. We have our two intermediates, D-glucose and D-fructose. As we saw, it is very simple. Just take one C double bond O unit and 5 COH units, combine them, and placing C double bond O unit on position 1 forms glucose, and placing C double bond O unit on position 2 forms fructose. Now what's with the D? It is related to the stereochemistry. We have drawn here the structure of D isomers because it is the most common form found in our body. Without going deep into the stereochemistry, for drawing D-glucose and D-fructose, you should remember that you keep all the O groups on the right side, except on the third carbon as we have drawn here. There is one more thing though. These sugars do not exist in straight chain form in our body. They rather exist in the cyclic forms. So let's draw their cyclic structures as well. For cyclization, carbon containing C double bond O forms the bond with the 5th carbon or semi-last carbon. A water molecule is removed and an ether linkage, that is COC linkage, is formed between the semi-last carbon and the carbon containing C double bond O group. So in glucose, it is formed between carbon 1 and 5, and in fructose, it is formed between carbon 2 and 5. And all the carbons in the middle are included in the ring. So glucose forms a 6-membered ring with carbon 1, 2, 3, 4, 5 and the oxygen atom, while fructose forms a 5-membered ring with carbon 2, 3, 4 and 5 and the oxygen. Again, for the D isomer, the OH on the third carbon is pointed only upwards along with the carbons left outside the ring. Rest all the OH points downwards. And these are the two 6-carbon intermediates of glycolysis. Now let's move on to 3-carbon intermediates. We will take 3 COH units and combine them in a straight chain. This forms a very famous molecule, the glycerol. And our intermediates are derived from glycerol. Let's see how. Alls or alcohols on oxidation gives aldehydes or ketones. If we oxidize the carbon number 1 on glycerol, we will have an aldehyde. And this aldehyde formed from glycerol is called glyceraldehyde. And it is one of our intermediates. If we oxidize the carbon number 2 on glycerol, we will have a glyceryl ketone. But it is not called glyceryl ketone. It is a 3-carbon ketone which is called acetone, and it has 2 OH or hydroxy group attached to it. Hence, it is called dihydroxyacetone. And if we see, glyceraldehyde and dihydroxyacetone are isomers to each other. Same molecular formula, but different structural formula. So, dihydroxyacetone forms our another intermediate. So, if glyceraldehyde is further oxidized, it will convert into acidic form, glyceric acid, from which if H plus is removed, what remains is glycerate, or another intermediate. So we have three glycerol-based intermediates: glyceraldehyde, dihydroxyacetone, and glycerate. We have two more 3-carbon intermediates, and these are derived from a 3-carbon alpha keto acid, which is pyruvic acid. That is CH2, C double bond O, COOH. If H plus is removed, it forms pyruvate, the final product of glycolysis. Pyruvate is a keto compound. Thus, its enol tautomer also exists, which is CH2 double bond CO minus COO minus, and it is called enol pyruvate, which is our final intermediate. So we have seen the structures of 6-carbon intermediates, glucose and fructose, and 3-carbon intermediates, glyceraldehyde, dihydroxyacetone, glycerate, pyruvate, and enol pyruvate. But these were the basic structures and names. In glycolysis, these exist in their phosphorylated forms, and to name them, only prefix such as 3-phosphor, 2-phosphor, etc., or suffix such as 6-phosphate, 3-phosphate, etc. are added. But the base name and structure remains same. Only difference which comes in structure is that OH becomes OP. So consider them as equal to identify the basic structure. This was all about the general idea of intermediates. Keep them in mind. And in next part, we will see how to learn and write 10 steps of glycolysis conceptually with these structures.
And thanks for watching. See you in the next part, and do not forget to subscribe for more.